All right. Hello, internets. Welcome to the Onco Data Podcast, one of our two podcasts here in the LARVOL universe. Back on after a couple weeks off, but we're going to get back in the groove. We're going to talk today with our most frequent guest, and frankly, I'm going to just call him a co-host at this point, Darko, to talk a little bit about what we're doing in AI and then network meta-analysis and all the incredible things you can do with synthesizing data. So that's sort of the preface for today's conversation. We will be having some more forthcoming podcasts. To everyone who's interested in this domain, we have World Lung right around the corner. So you're going to see probably a couple podcasts from a couple of us at LARVOL about World Lung and the data coming out of that, especially oncologist reactions. And then before we know it, it'll be ESMO. There's also IML, myeloma meeting coming up. So we're going to have a lot of things coming out on social. So I'm just doing a little bit of an advertisement. That stated, Darko's back with me, and happy to have you, Darko, who's not just a guest and not just a friend, but also intimately involved with our work in network meta-analysis, our internal endeavors in biostatistics and synthetic data. So that's what we're here to talk about today. So Darko, I think I gave you a rather long introduction, and frankly, I don't know if it's needed, but if you wouldn't remind people about yourself and some of the work you do, Darko, and we could dive in.
Thank you so much, Mark. It's really amazing to join you and really amazing to earn a title of almost a co-host.
I think we're pretty much there. Yes, sir.
Really amazing. Thank you so much, Mark. And just a very quick intro. You made a very good beginning of the intro. So I'll just give some information for the audience about myself. I'm someone who is a biostatistician for a very long time, but also someone who works in the field of artificial intelligence, creating AI models, and so on. In terms of my collaboration with LARVOL, there are a number of projects that we're working on together, and I assume that we will be mentioning some of them today, and really looking forward to an, I'm certain, interesting discussion with you.
Absolutely. Well, then I'm just going to go right in and put you on the spot, Darko, because two things I wanted to pull up on the screen that we talked about before, but wanted to pull that up and give a quick demo, and we can talk about where things are going. So I'm going to start with pooled analysis as soon as I can get my share screen going. Let's go over here. Let's see. Let's go to... All right. Hopefully my screen is sharing, Darko. Can you see that?
Yes, I can see your screen. Yes.
Awesome. So obviously we're on LARVOL CLIN right now, clin.larvol.com. If you're interested, you can give it a free test run. But today we're going to be talking about maybe what you might call some of the advanced features. And as I said, I wanted to start with pooled analysis, just quickly show a demonstration of what it can do and talk about some of the features that you might see soon on CLIN. So what I'm actually going to do over here is go to our digitized Kaplan-Meier curves. We have many thousands of these across our our universe of 100,000 plus trials in CLIN. And I was actually just looking at breast cancer, so maybe I'll stay on that theme. And I don't know, maybe I'll think of a newer or maybe an older trial, and I will go ahead and fire that in here. So something I was looking at earlier, because I was just asking a bit of a random question about where you have a estrogen degrader. And I was just thinking, as a former scientist, what does that look like with a PI3 kinase inhibitor versus a AKT inhibitor, because they're in the same pathway. So that's why I was looking up BEL-2 and also looking up this Capitello trial, if I'm pronouncing it correctly. So that would be 291, actually. And so what I'm going to go ahead and do, and then I'm going to turn over to Darko to see where we're taking the software next. But just to illustrate the point is I'm going to go up here to Kaplan-Meier, and I'm just going to choose PFS for this one trial. And I'm going to populate this up here, but I'm going to do a quick changeover. And I'm going to select for pooled analysis. So right now I have PFS for this Capitello 291 trial, which is newer data within 2024, and I pulled PFS as I stated. And then I do have PFS data from this older trial here. So this dates back to 2017. And so what I've done here is put those in one just overlay, just one multi-curve visualization. And then what you can do here is actually run pooled curves. So I'm going to go over here to this button and make sure I'm describing this correctly. But basically what this is taking is synthesizing, if I could use that term, those curves using statistical methods for which Darko is an expert, and creating it from the original Kaplan curves that I showed. So that was a bit of a demonstration, but maybe Darko, what points do you think as of the current software did I miss highlighting? And what are you and the data analysis team working on next for pooled curves?
Yeah, I think that was really a great explanation and really a great example of how pooled analysis can be useful to analyze a specific biological pathway in relation to the survival data and the Kaplan-Meier curves we have. So in this case, what's very interesting is that, of course, that we are taking multiple trials here, and we are taking multiple KM curves to try to bring them to the common data model. So that's really one of the biggest obstacles in the oncology research when we are speaking about survival data and Kaplan-Meier curves, is that there are so many different curves, so many different trials, so many different analyses, and it's not easy to bring them to the common data model. And common data model is needed to perform any form of large-scale analysis. So really the goal of this pooled analysis when we built the product was, can we go large scale? Can we take many, many trials, as you did here, multiple trials, and then create one pooled curve out of those multiple trials? Now, to do that, we need to create a common data model. We need to have similar intervals, practically same intervals. We need to adjust the data so that data for each curve is on the same scale, have the same starting point, and eventually turn that into a common data model. Now, as you mentioned, when you were creating these curves, to do that, we need to use some form of data reconstruction, right? So we are reconstructing the individual patient data curves. To achieve all this that I was speaking, we need to create individual patient level data, right? We need to go from study level data, from study level digitized curves to individual level patient data. So we are reconstructing individual level patient data using specific statistical methods. And effectively what we are doing, we are reconstructing synthetic data from original curves as accurately as possible, and then utilizing that for a variety of potential use cases. So Pseudo-IPD is just the beginning. So this is already a feature that has various use cases. One of them, which is obviously very interesting, is that you can take many, many different trials and construct a super KM curve, right? So this was impossible, some time ago, virtually. So right now we have this capability, and we are constantly adding novel statistical methods into the workflow so that we are addressing all the problems that may exist while reconstructing so many curves into one giant curve, right? So we're discussing proportionality of the hazard. We are discussing censoring, different censoring types in these curves, and we already have many solutions how to equalize all these. So for the audience, it can be seen that a lot of problems that we resolve here are revolving around this common data model and individual patient data, right? So that's really the core of this technology. And then on that core, we can build so many different products and so many different use cases. So this was really a fantastic example that you're showing where you have many different curves. And if you're interested to know, hey, if we were to somehow merge all these together, what would we get? We're effectively conducting a large-scale meta-analysis, but the difference is that in this type of meta-analysis, we have the temporal patterns. We can see what's happening with this giant curve. We can see the patterns when the therapy starts. We can see the shape of the curve, which is as we add more and more data, more and more curves, it starts to look smoother, right? So we're going from individual curves which have this stepwise sharp changes to a smoother statistical curve that now has more statistical power, obviously, because we have much more data, much more samples, patients level data included in the curve.
I was actually going to go to, well, I was going to go to network meta-analysis, but I sort of want to sit here for a second on pooled analysis. And as I was going through and I was demoing, just wanted to point out that you can select, for example, you can do a series of pooled curves just for control arms and versus just for maybe experimental arms. And you can actually do that in conjunction, well, not conjunction, but in parallel. So taking a number of experimental versus a number of control arms. I just wanted to call that out from a practical perspective. But from a statistical perspective, I guess my questions is, let's just focus in, well, we have, these are two experimental arms for the comparison I was making. Let's just say for sake of illustration, let's ignore the pooled curves. Excuse me, let's just take two curves. I'm just going to do this conceptually, Darko. If you have two pooled pseudo-IPD curves that you're trying to do the typical analysis on that you would have for, quote, real curves, what is possible and what's not possible? Obviously, a curve can have a median, but obviously you could have a synthetic hazard ratio as well. And I think that's maybe a next step. So I guess I'm asking for anybody who would like to do this type of pooled curve analysis, what type of statistical methods are at their disposal, and then what's not. Does that question make sense, Darko? You see what I'm saying about this whole construct of pool curve comparisons?
Yeah, this is actually a very good question, and it aligns with what's next for this technology potentially. So when we go large scale like this, when we create either one or potentially multiple pooled curves, as you said, so we can create two pooled curves, like one pooled curve for the intervention arms, for all the intervention arms we have, and one pooled curve for the control arms. So obviously, all the biases that exist in the individual curves now start to pile up, and we have a compounding effect of these biases, right? So many statistical methods which would normally be possible for, let's call it the normal KM analysis, would not be feasible on this large scale, because all these biases, they would break a lot of statistical assumptions that exist, such as proportionality assumption we were mentioning before, censoring can be so different. Many hypothesis tests, such as log rank test, for example, they cannot be performed in such case. So what we are doing right now is we are creating a foundation, a framework, and the more news on this, I think, will be communicated very soon with our audience. Is that we are creating a robust statistical system that will deal with all these problems and enable things like creating two pooled curves. Each pooled curve could have 20, 30 studies worth of KM data. And then we compare them the same way we would compare them in a randomized control trial, except this is not a randomized control trial. This is a different approach. But we are creating that approach where someone can, for example, calculate a hazard ratio and then also do a special statistical test. So I won't be revealing more about that test until it's actually deployed, but we are making a substitute for the log rank test, which wouldn't work then. And we are creating a test that can give user statistical significance in addition to hazard ratio, in addition to confidence intervals. So we are really doing a novel approach to the meta-analysis space. I would say this is a very unique way of a meta-analysis where we actually have the temporal data and patterns, and we can perform, the proportional hazards models and
Sure.
everything which would not be possible on this large scale, right? It would be disregarded just, I don't know, a few years ago. This would not be possible. It would be disregarded as not valid, right? But now we're entering this space where we're finding the solutions for the problems that existed before. Now, the main use of this technology, again, is going large scale for now, just going large scale. And that's really important for the oncology, right? Where statistical power is vital, right? Large statistical power can enhance decision-making process, oncology intelligence for companies, and for researchers doing large-scale analysis. I think that's a set of very important approaches that are enabled with this pseudo-IPD technology. I mean, we are calling it the pooled analysis, but really it's the pseudo-IPD technology that's behind the pooled analysis.
Absolutely. Well, I wanted to sort of, obviously there's a lot of downstream applications to this that I guess I'd planned to ask you about, whether it's synthetic data. We're speaking to synthetic data. That was in my notes to ask about, but that's what we're speaking to. There's certainly that. There's digital twins as a possibility. And then there's network meta-analysis as another topic, which is another aspect of the software that we built. But I really just wanted to drill down on, pooled analysis, but you're correct to say it's about pseudo-IPD technology to just really level set with anyone who's interested about what are the limitations of it, what you can calculate and how to make valid comparisons, and what you can't do. So I guess putting all of that out there to be transparent about what's possible and what's not is sort of why I wanted to ask some of those questions today. That being stated, we've reached, I guess we're coming up on time in the world's shortest podcast here, the Onco Data Podcast. So I would like to thank my 99% there co-host, Darko. Maybe another one of these, you're at 100%. So thank you, Darko, for joining me today.
Mark, really an interesting discussion. Thank you so much for having me, and hopefully I will reach that 0.1% to be the co-host.
All right. Well, we'll see if we can get you scheduled and get you to be 100%er. So thank you, Darko. Thanks everybody for listening.
So much.
Follow us on social media. Cheers.
Cheers.